A very good evening, everyone. A warm welcome to all of you for today's exciting live session hosted by the BioLoom Islands, the first ever synthetic biology company where innovation meets the future. We're truly thrilled to have such an amazing audience joining us from different places, and thank you for taking the time to be with us today. Today's webinar, Tomorrow We Rethink, is more than just a talk. It's an initiative from the BLI Think Lab, a space dedicated to exploring new ideas and trying new things for the future. It reflects our efforts to challenge existing concepts and rethink the way we understand biology. Tomorrow We Rethink, a thought provoking exploration into how biology will never truly fixed and why it's time we start rethinking it. This session will dive into reimagining classical DNA synthesis through emerging DRT3 and DRT7 mechanisms, opening up new perspectives in synthetic biology. And now it's my absolute pleasure to introduce our speaker today, Dr. Lalinka Herath, CEO of the BioLoom Islands Private Limited. Before we begin, just a quick few reminders. Please keep your microphones muted throughout the session, and if possible, we'd love to see your faces, so do turn on your cameras. All right, without further ado, let's get started. Dr. Lalinka, the floor is yours.
Okay, thank you very much, Kavya, for the introduction for our session, and also very good evening, everybody who are joining the today's session. So the today's topic is going to be a little bit different one from it's not going to be a conventional biological science topic that we are going to kind of discuss in today's session, everybody. I don't want this session to be another talk, but an interactive sessions that we can, of course, share our ideas, our thoughts on this uh, particular aspect in biology that we are going to discuss in today's session. Um I can see some of my students joining as well. Sanjula and everybody, do you guys want to join the discussion as an interactive session? I expect you guys to turn on the cameras as well. Then we can have a very effective session, right? Are you guys around? Until, yeah, you're prepared. Can I know the everyone joining today? How many of you are from the molecular biology background? I mean, not the heavy molecular biology. Uh, like at least if you know what DNA means and how DNA replicates, can you guys please put your hands up if you're from molecular biology background? Hands up in the sense, nice. Yeah, we have Shajwa, yeah. Right. Everybody, I'm waiting for you all to. Wonderful. So we are here to because we are excited about this new insight uh, on DNA as a molecule and how it behaves, right? We know what we learn in our universities, maybe in our conventional thinking about DNA, how DNA as a molecule, how it behaves, right? So how many? How many? Let me check. Others are not from molecular biology background, guys. At least you learn the DNA replication, right? DNA replication as a mechanism. Sanjula, do you know DNA replication, how it happens?
Yes, sir.
Right. So I want to have a kind of effective conversation between you guys because I'm asking a question here. Biology was never fixed, so why are we? Right? So the first question I need to ask, is that true, everybody? Biology was, yes, I'm from molecular. Perfect. Yeah. So we are here because we are excited to understand how the biological systems behave, right? Um, and change the way we look at the biological systems as molecular biologies. Right. Uh, okay. So I'm asking this question initially. Biology is never fixed. So why are we? Was it fixed or not? Can I have some answers then? Was biology fixed? Huh? Are we fixed as human or are other animals? Are they fixed? And the plants as biological system, are they fixed, guys? Right? So that's the question we initial. Also, we are evolving slowly. Perfect. Yeah. The biological systems, we are all biological systems, right? We keep evolving, we are adapting, we find strategies to evade our threats, right? So it's all about it's a survival game, right? So we are trying to survive. Yes, nice. Yeah. No. So why are we now thinking about biological systems, the way we learn molecular biology, which are the advanced aspect of biology? So do we have to look at biological system in a way as those are static bodies there? So we can, of course, this evening have a discussion on this and maybe to change or maybe to slightly over or slightly to alter the way we look at biological systems and understand the way we learn about these molecules in biological systems, right? So that's the idea of this particular session. Uh, Wait a second, let me share my screen. Not this. Before that, I want to share the famous quote by the great scientist Erwin Schrödinger. So he's a great physicist, right? Uh, and my personal favorite scientist, guys. He always says the task is not to see what has never been seen before, but to think what has never been thought before about what you see every day, right? So learning, looking at biological systems and understanding biological systems also. It's about not that we are not seeing things because as humanity, for maybe thousands of years, we have seen everything around us. But the question is, have we thought the certain questions? Did we ask the questions so far as the humanity that we want to ask to understand biological system? So today's session also, I want to focus on this aspect and then see how biological systems at molecular level behaves, and then via one of these emerging and one of the very fascinating recent outbreak in the fundamentally very interesting outbreak we recently came across in molecular biology world, which is the DNA polymerase. As a molecule, we know that's the molecule which synthesizes more and more copies of DNA, but we have recently come across new family of molecules that, of course, make DNA. Making DNA is a very interesting process in our biological systems, information copying, right? The DRT3 and DRT7 are new group of molecules which follows an interesting mechanism in order to make copies of DNA that we were not previously believed. We were not believing that DNA could be copied based on without having a template. Guys. But we are trying to understand how certain molecules can also behave differently in order to survive the certain threats that they face in as to sustain as biological systems. Okay. Uh, so this is about fundamentals in molecular biology, guys. We have to understand bit of fundamental molecular biology. I know all of you have learned this thing, the main idea of making DNA, right? In nucleus of cells to make new DNA, what needs to be done? Sanjuna and others? Pramisha. So what's the process of making new DNA? This is something we all learn in our universities, right? As one of the first topics in molecular biology, biomolecule topics, how DNA, new DNA can be made inside of our cells.
DNA replication.
DNA replication is the process. Thank you. Right. So how it happens then? If you want to quickly recall what to traditionally learn about making DNA, what are the components required in making new DNA copies? Hello. Right? DNA can be made by based on information in DNA, which means it's more like a photocopy information, right? DNA is being copied. So we need a template and an enzyme, which is DNA polymerase. Another mechanism we observe in biological systems, guys, which is making RNA based on DNA, that you call the transcription process, right? This is the way of transforming information on DNA onto another nucleic acid, which is RNA, and it is to be transferred to protein, if anything, something to happen in cells, so that we learn in our fundamental topics. And another interesting process that we observe in biological systems, DNA can be made based on RNA, right? So what do you guys call this process? In that case, making cDNA, complementary DNA based on RNA, right? The RNA template and reverse transcriptase is the enzyme in that process. So everybody, look at these three mechanisms. You can see some common feature in these three mechanisms that we learn in our conventional classes, right? In molecular biology. So what are the three common, what's the common characteristic? Huh? Someone tell me.
It's template dependence.
So this is, yes, perfect. Like it's template dependent because DNA or any nucleic acid so far that we've learned, so they cannot make new nucleic acid without referring to information in a reference or a template. They have to have a template in order to make new DNA, right? So basically these are all chemical reactions, guys. But if you can understand DNA polymerase, RNA polymerase, and reverse transcriptase, these are enzymes which are catalysts, reaction catalysts in biological system. Basically, making new DNA is the product, right? To have nucleotide as the substrate, right? So substrates are nucleotide, you have DNA polymerase, and then you get more and more DNA as the product. So it is an enzyme catalytic reaction. Here also making RNA based on DNA. Another mechanism. Making new nucleotide and there's an enzyme involved. And here RNA is considered as the template, and based on RNA's information, cDNA can be made. Again, it's a template dependent because you need RNA molecules information to make cDNA. So the common feature here that we observe, so it is template dependent synthesis of nucleic acid. right? So in your classical molecular biology topics, you use this single principle. So we never thought that new nucleic acid can be made without having a template. You remember that you learned that DNA strands need to be separated and DNA polymerase should be bound, right? And then it should copy the information on the template strand to make a new DNA molecule. It's template dependent information transferring process, right? This is done by these classical molecular biology topics you learn about these conventional methods, right? So you know that this synthesis happens in the direction of 5' to 3', 5' to 3'. And it has to follow the Watson-Crick base pairing rule. What is Watson-Crick base pairing rule? Someone? Let me know. The Watson-Crick base pairing rule? Can someone? You'll get, yeah, Ratha, I don't know how to pronounce it. Ratha Rajan. Yeah, Rajan, you can just unmute your microphone and let me know your answers, even others, right? So this, then we can, right. Yeah, it's the rule that nucleic acid needs to follow in making new molecules. Only certain bases can be base paired with some complementary types of bases. What are these complementary bases? It's adenine to thymine and guanine to cytosine. There are 4 bases, if I'm correct. Is it correct, guys? How base pairs in DNA? Hello?
Yes, sir. Adenine to thymine and guanine to cytosine.
Okay, Shihara, is that correct? Hello? Any other answers, guys? Is it adenine to thymine and guanine? I don't remember. Himal, what's your answer?
Yes, it's correct.
You sure?
Yes, DNA, adenine to thymine, and guanine to cytosine.
Right, so that's the rule, right? Then if you say so, I just believe you. So then we have to consider that Watson-Crick just told us it has to happen in a way that adenine should go base pair with thymine and guanine should base pair with tyrosine, ATGC, right? Right, there's another classification, right? The purine, pyrimidine. So those things that you are supposed to memorize as molecular biology students, right? Okay. Now, the thing that I need to highlight, I wanted to highlight in this case, all these processes that we learn making DNA based on DNA, which is the DNA, the replication process, you need a previous DNA molecule to provide information. The process transcription, making RNA based on DNA. So in that case also, we need information on DNA to be copied onto an RNA molecule. Making cDNA based on RNA, very interesting, guys, but still not that exciting, because to make cDNA, we need the information on RNA. Again, the process is template dependent, even though the information flowing direction is different. So it actually flows information from RNA to DNA. So that's something that we can see there's some excitement about violating or kind of violating certain rules, right? But still, the process is template dependent, right? Now, moving on to These are the three main DNA making processes that DNA nucleic acid making processes that we learn in biological systems. Right? The common feature is template dependency. Right. So let's see, these are all molecular machinery. So these molecules work inside our body as machines, that they are intelligent machines that perform certain function. The DNA polymerase was one of the very interesting molecule. The very fascinating, the way the DNA polymerase makes new copies of DNA. This is an enzyme at the same time. It's a multifunctional molecular machinery. It's more like a robot that performs multiple functions in making a new copy of DNA, right? So you think about certain things that a polymerase, DNA polymerase molecule as a protein, three-dimensional protein perform. So they can actually bind onto a DNA template, right? There's this catalytic activity is the core function of DNA polymerase molecule. So it can bind to single-stranded DNA. So now you have to visualize this, what you learned in your classes, guys, in your mind, then you can picture. There's these strands. I know all of you have learned these things, right? There's these DNA strands, and then it has to be, yeah, DNA. I'm trying to draw some molecule. Is it correct? Wrong. Right. How do you learn about DNA? So it looks like something like this, right? What's the shape? Double helical structure, right? DNA needs to separate its strand. Okay. So I'll just draw it like this, but you have to know that it happens in a different manner, three-dimensionally. Let's say these are bases. Phi dash, three dash. Again, 3 dash to 5 dash, right? From which direction the polymerization we were? Hello, guys. Can you help me out here? The polymerase should bind here if it is to make molecules, synthesize DNA in this direction, right? Then this become 5 dash to 3 dash, right? And on the other strand, it goes in other direction. So I always ask a question, are there two polymerases involved in this process, guys? Because there are two strands. How this actually happens? How to take this line? I got it stuck. Can you guys hear me?
Yes, sir, we can hear you clearly.
Yeah, but I can't take this line. I don't know what happened. These chats. Let me stop sharing and come back. Okay, right, you're back. Yeah, the polymerization happened in 5 dash to 3 dash direction. So it is a template dependent mechanism. Single strand should provide information for polymerase to incorporate new and new, more and more incoming nucleotides into a drawing strands to make more copies of DNA. So this reaction, you know that it's basically makes phosphodiester bonds to merge two nucleotides, right? Uh, so if you look at it about the reaction with chemistry, so there's substrates in this reaction which are DNTPs and magnesium 2 plus ions as cofactors to support DNA polymerase to synthesize the strand. It happens in 5' to 3' direction, right? Basically, the mechanism is the polymerase read the template. Polymerase as an enzyme should bind to a DNA strand and then it should read the information while moving across the DNA strand in order to make a new copy, guys, right? So this is an enzyme catalytic reaction to make phosphodiester type of bond. Do you guys know what are phosphodiester bonds? How many of you all know how the phosphodiester bonds form? Right, can you guys, raise your hands? The actual chemical reaction of making it that to draw, right? In making, incorporating new molecules, nucleotides, right? Okay, right. It's basically a reaction. And when you think about this molecule, the DNA polymerase used to be a very interesting molecule that amazed a lot of scientists because how accurate the mechanism of DNA polymerase making new strands. It can perfectly read the information on the template strand and then only allow the incoming nucleotide should be selected that is to become complementary to the template strands. Otherwise, it won't let other nucleotides to come and bind. So that is governed by DNA polymerase molecules multiple multitasking ability. It won't allow the other base pairings, but again, it has proofreading ability as well if something wrong incorporates into it. But the mechanism used to be something template dependent, as we discussed. So it always rely on the information on the strand, DNA strands that provide information. So if there's C, it calls for time, right? So that happens across the it dependence. Basically, the DNA polymerase depends on the strands too, because it doesn't have the memory to make their own DNA. So it's always dependent. Again, we will move on to the other molecule, other mechanism, the RNA polymerase machinery. So we can call them molecular machineries because they are working as miniature factories of small machines, small robots making small molecules, right? So the RNA polymerase as a protein assembly, the key features of RNA polymerase and how it differs from DNA polymerase is that you don't need primers. You know, in making DNA, the priming, initial primers should attach to DNA strands. Then it will elongate towards the 3' direction, 3' direction to make new copy. But RNA polymerase does not need a primer to initiate the nucleotide making process. No primer required, right? It happens de novo manner, which means it can do it by its own. But the information should be retrieved from the DNA strand. Promoter recognition happens with RNA polymerase. Some new ability. Comparatively, you can understand what are the other advanced features of RNA polymerase in compared to DNA polymerase. It can also, it doesn't need primers to make RNA strips, right? Promoter recognition directs this promoter, which is the area that RNA polymerase land on DNA and then start making an RNA transcript. So these are just something you learn from auto recognition happens, and then you know the initiation site and RNA transcript are being formed. Transcription bubble unwind. So that's the mechanism, guys. Uracil will be there in RNA molecules instead of having thymine in 4 bases, right? Uracil replaces thymine in RNA. So these are the features of RNA, right? Even though it differs from DNA, this mechanism is also template dependent. Did you understand that, guys? This fact that all these mechanisms are dependent on their template is very important for us to understand what we are going to discuss today in our new mechanism. Everybody? Not only this, now look at this molecule as we initially summarizes, that this is it's very interesting molecule, the reverse transcriptase machinery. How many of you all have heard about reverse transcriptase? Hello? Reverse transcriptase as an enzyme that can make have you heard about reverse transcriptase enzyme? What does it do? Sanjula, yeah, explain me what happens with the
Simply, it's like make DNA from an RNA template.
right. So say that again. Starting
Uh,
from RNA, it makes
yes,
DNA, right? Okay.
yes.
Okay. What are the interesting features in this molecule? The certain enzyme use. The reverse transcriptase is the enzyme, right? Of course, it's template dependent, right? Is it correct, guys, about reverse transcriptase?
Yes.
It depends on template in making DNA. Even though what happens, the process is reverse in making nucleotide, still the information on RNA provides guidance to make new DNA, which is known as cDNA synthesis, right? Himal, do you want to explain this mechanism to us?
So that means the making cDNA from the RNA.
Yes, yes. Can you guys let me know how it happened?
So it basically mRNA. From the mRNA, mainly this is used by retroviruses which have RNA as their genetic material. So when they want to uh, make the new DNA, so they have reverse transcriptase encoding gene.
Mm-hmm.
So they encode reverse transcriptase, and then on the mRNA they produce cDNA, and from then cDNA again they make mRNA and translate the proteins. Likewise, so that's basically found on the viruses, not animals or plants like us.
Yeah, viruses uses this mechanism, right?
Yes.
Imandi, is it correct? The mechanism Himal explained? Hello?
Because I was lagging. I couldn't hear it good.
Yeah. Can you explain the mechanism here with retroviruses that use making reverse transcription process?
Reverse transcription is the DNA, RNA is converted to DNA, So give me a minute.
Okay, right. Anybody else who wants to explain this process to others? Have you learned this in your classes? What happens here? Right, let me see. So there's RNA split, there's RNA split, RNA single stranded, right? Okay. And based on the information on RNA split, the reverse transcriptase enzyme can make a single copy of another copy of DNA, right? So it's I can't draw the helical thing here. Right. Another copy of DNA. Is that what happens initially? So you want to recall what you learn in order to understand how these molecules behave, right? So this is like the light strand is a DNA. This is an RNA, right? Now, see what happens. The template, the RNA makes something called complementary DNA based on RNA's information. Then RNA's H degrades RNA. It's an enzyme, right? Thus, another enzyme can degrade to a RNA molecule. Now you have DNA copy, right? And then again, another polymerase or reverse transcriptase can bind onto this molecule and make another DNA copy. And to make it fully double-stranded DNA. So this entire molecule you call cDNA molecule. Is that what happens here, guys? Right? So that's what as I am saying happens in this mechanism, right? Okay, guys. Now, even though this mechanism is different from making DNA, replicating DNA, right? Making DNA based on DNA, making RNA based on DNA in transcription. Here, making DNA based on RNA. So the direction of information flow is different. It's the opposite direction. But still the mechanism is template dependent. So that we have to understand in our mechanisms here. So this is again template dependent because the RNA template should be there initially in order to provide information for reverse transcriptase to incorporate bases. Clear, guys? Is that clear for you all?
Yes, sir.
Right, great. You don't know what happens. Okay. Right. So that's what happened, but the process is template dependent. In this case also. Right? Okay. Now we know three main mechanisms that making DNAs, RNAs in biological systems: replication, transcription, reverse transcription mechanisms that we will study. Okay. Now next, moving on to a new mechanism. So this is the interesting part. In this year, in 2026, a group of scientists discovered the molecule and other molecules that found in. It was found in the defense system of bacteria against bacteriophages. Now, someone explain me what happens. How do bacteriophages infect bacteria? Hmm? How can they infect bacteria? It's a war, combat between bacteria and viruses. Viruses want to hijack the DNA replication mechanisms in bacteria because they can't make their own nucleic acid uh, without the support of bacteria. So, can someone explain me how it happens? What does a virus do in entering into bacteria's body as a phage? Sanjula, I
Yes,
need
sir.
your
Yes, I'm here. So,
continuously do this because right?
yes, so like what happens when the infection happens, first they attach to the bacterial cell wall using like specific receptors, as I remember, and
Okay,
then they penetrate in and injects its DNA or RNA into the bacteria. That's how
okay,
they infect, and then it's get replicated in the using the host.
right. So how do they replicate in host?
Like so,
They use the
so the
host
page
machinery.
Steal,
yes, yes, they steal like it takes control of
steal.
the bacterial
Hmm.
metabolic.
Yes. Any other answers? Anybody? Yeah, I heard somebody else has explaining.
Yes, sir. I think they use the host machineries to replicate the proteins and the necessary enzymes.
Yeah, that's it, right, Vashik? Yeah. So they can sort of hijack, right, the machinery, right? Then what happens to bacteria in that case? Because they are like encountering some sort of stealing, right? Because they deprive molecules to synthesize their own DNA, right? In bacteria. Is that correct? Therefore, bacteria needs to come up with mechanisms, right? Yes, to fight against using host replication mechanism. Yeah, lytic cycle, derivative. Yeah. So thank you, Taran. Yeah. So they need to come up with mechanisms to act against this infection. They want they don't want bacteriophages to enter into bacteria and hijack their molecular mechanism. So to act against these infections, right? Transmission, bacteria come up with certain new mechanisms or strategies that can maybe destroy the viral nucleotide entering into bacterial cells. So, do you guys know how CRISPR-Cas system was initially discovered? You know, that's a popular tool, right? Very famous tool in applied molecular biology application. The CRISPR tool, it was discovered in bacteria as a defense mechanism against bacteriophages. So do you guys know that? Have you heard of it somewhere? Right? How CRISPR tool was discovered initially? How many of you will have heard it?
Yes, sir.
So it's as a part of the defense system, right? bacteria's defense system against bacterial phages. Now here also the DRT3 is a new molecule that scientists recently discovered as a molecule. It's part of bacterial antiphage defense system, right? So if we try to understand what happened here in this molecule is, so it's very interesting how it assembles, this particular molecule assembled in order to make certain copies of DNA segments. And it deviates from the theories that we learned previously about the DNA polymerase, RNA polymerase, and also what is the reverse transcriptase. All these molecules are template dependent, which means basically as a molecular machinery, the DNA polymerase, RNA polymerase, reverse transcriptase, what they basically do is that they bind to a nucleotide strand and basically copies the information on it. Right? It acts as a photocopy machine to make more and more copies. Right? We never expected DNA polymerase, RNA polymerase, and the reverse transcriptase to make new nucleotides, so new DNA, without having a reference material. Like in a photocopy machine, you have to insert the original. Then based on original, you get more and more copies. So you can think of the photocopy machine as the maybe the enzyme, the DNA polymerase, RNA polymerase, and the reverse transcriptase. You have to input the template, otherwise it won't make copies of it, right? Now, how interesting this molecule, DRT3 molecule, fundamentally in molecular biology, guys. And it's mind-blowing what it does at certain point as a molecule, right? So now, first you have to understand the assembly of this molecule, how this molecule looks like, right? To simplify that, I can draw some diagram. DRT3 is a molecule. Now look at this explanation. So it says DRT3A. It's as a full molecule. I have problem when I pick the annotation. I can't see where's the arrow goes or anything. So I must. That's why it takes time for me to switch. Right. Now let's say it says DRT3 molecule has three main components. Now I'm going to have to annotate on the slide. You can focus only on the slide. It says it has a DRT3A component. Let's draw it here. It's a protein component, which is DRT3A, right? And also it can act like RT-like protein, which means reverse transcriptase-like protein. Okay. And another component of this molecular machinery is that right. There's another component, which is DRT3B, right? It's a second RT-like protein, right? So let's first draw the molecule in three-dimensional form. So it says it has another component, which is DRT3B, right? Let's draw it like this. So that's DRT3B component. And then there's another part. NC non-coding RNA. You can just draw the non-coding RNA component as said. Non-coding RNA. So that's a non-coding RNA. Now this certain unique assembly exists as a hexamer form, because which means you have 6 copies of this assembly in DRT3 protein. Okay. Basically, you have 6 copies of this assembly. Right. Now, this 3 component can perform a very interesting mechanism in making more and more new DNA segments in bacteria as a bacterial antiphage defense mechanism. So what's exciting, what's interesting about this mechanism is that it violates one of the fundamental rules. Not that it violates, it preserves the rules, but it expands its boundaries towards new fundamental insights about how these molecules can behave inside of biological systems. Let me explain that interesting aspect here. Now, refer to the mechanism here. The DRT3A. So it says it's an RT-like, which means reverse transcriptase-like protein uses non-coding RNA as template to synthesize poly-GT strand. What does that mean, guys? Anybody wants to tell me? Huh? So we are trying to understand one of the recent breakthroughs in the entire biology, history of biology, guys. Who wants to explain that to me? Someone wants to unmute and explain that. It says the DRT3A domain, right? Globular component uses non-coding RNA, which is indicated in green color here in my, we could say it's not a proper drawing, guys, right? As template to synthesize poly-GT strand. Who want to explain that part? Someone, please. So, because this is an interesting mechanism we are together trying to understand, recently discovered in biological systems. Hello. Who wants to give it a shot? Anyone? Hello. Someone help me out. Sanjula, did you study this?
So like DR3 means like defense associated reverse transcriptase. So what they do is they like synthesize poly GT. So
Yeah.
poly GT means like GT sequences.
How does it?
So it's in the non-coding RNAs, they are having like specific motif sequences, the motif with the ACAC.
Right. Okay.
Poly AC. So from that they are using, they are synthesizing a single-stranded poly GT.
Right. So which means alternatively repeating units of GT stretch can be made, right? Based on the information on the non-coding RNA segment indicated in green color here, right? Am I correct?
Yes, sir.
Let's say this is the non-coding RNA. Can you see this annotation?
Yes, sir.
So let's say this is the non-coding part. So it has alternatively, what should it carry?
ACC.
Cytosine CACA, right?
Yes.
For poly-GT, right? So basically it should contain CACA segment, right? Based on that information, it can create the DRT3A component of the DRT3 molecular assembly machinery. What it basically do is, by using non-coding RNA information on this small stretch of RNA, right? I can't find that. This is how I'm going to find the. Do you understand my pain, Sanjulu? I can't locate the arrow once it.
So, can you like use the laser pointer or something?
Yeah, for drawing, how can I do that? I don't know what's wrong with this.
In
But
here,
that's
like
good.
using the pen, like the pen.
Right. Oh, it doesn't work here. Laser
Here,
point.
laser.
Ah, it works. It works. Yeah. So I can pop the laser point, but in annotating pen doesn't show any colors. Laser pointer works. Right. Right. Now look at this. The DRT3A depends on non-coding RNA stretches information in order to draw what? In order to make here poly-GT stretch. Okay, poly-GT stretch. Did you understand, everybody? Right? What happens with DRT3A component? It makes a poly-GT stretch. Can you understand? Basically, this can make a poly-GT stretch. Poly-GT. GT alternative stretch of DNA, right? And this is the non-coding RNA part indicated in green color here. So it acts as a template, right? Now what DRT3A segment does here, is it exciting, guys? What's the hype about this molecule? Is that component, what it does, is it exciting? Everybody? Hello? Anyone? Sanjula? Others?
Yes.
Are there? Yes?
Yes, it's using an uncoding RNA.
Right. What about others? Right. Thank you, Sanjula. Ihan, is this exciting as a mechanism? What DRT3A does?
Yeah, I think so too, sir.
Right. Yes. Who else? Anybody else? Any other answers?
So why is it actually called a non-coding one if it is, coding for the DRT3A?
Yeah. Who's that? I can't figure out who asked the question.
Vasit.
Vasik. Yeah, Vasik.
Yeah.
It's called a non-coding RNA because it's not involved in the mRNA. It's not an mRNA type. Basically, it's an RNA scaffold, right? The thing is, it is attached as a part of this assembly. The ncRNA component is there and it supports in making a poly-GT stretch, right? Okay. Can you all understand that?
Yeah.
Right. So now I ask a question. If this mechanism exciting, because we are here to discuss how interesting the DRT3 molecule behaves in making DNA, right? Uh, right. Sanjula, for you, it's very interesting, right?
Yes.
What about you and for Yehan? What about Vashik? What do you think? Huh? Hello. Others like, shall I random? Himal, what do you think? So we are trying to discuss something here, right?
I think it's actually interesting how DRT3B synthesized the polyacetal without using a template.
Really, I didn't even look at it. Is it right? Why not? Why not DRT3A component?
Um, so actually, it is using a template already. We know template using machinery. So
Even for me, of course, it's interesting, but still, it's template dependent, right?
yeah,
Sanjay,
that's why.
hello,
Yes, sir,
Sanjula.
that's why. Okay.
Yes,
Yeah,
sir.
yeah. The DRT3A component still template dependent. So,
Yeah.
it's not that intelligent. Okay, TNA trying to copy and making a GT copy based on what it's got from the non-coding RNA component, right?
Yeah.
So, classical molecular biology on DRT3A, right? Correct. Huh? Imandi? Imarshi? Is Imarshi Imandi around? Are they around? Who is? But yeah, I don't know most of the names of this. Pramisha? Pramisha Rajpaksha. Pramisha,
Yes, sir, I'm here.
what do you think about these mechanisms, the DRT3A components, what it does? Because we talk about DNA polymerase, RNA polymerase, reverse transcriptase, and now we are looking at a new molecular assembly, which is DRT. And part by part, we are trying to figure out the mechanism of that molecule, how it makes a DNA component. DRT3A component makes, it says, make the poly-GT stretch based on the template of non-coding RNA, right? So what's different in it when it compared to the classical molecular biology mechanisms we learned? Of course, it's reverse transcriptase is somewhat unconventional, but still it's template dependent, right? Now we are seeing something very interesting that we have never seen before about proteins, guys. When you look at the DRT3B component of on the DRT3 molecule, right? So we have, it says the second RT-like protein, which is DRT3 segment, synthesizes complementary polyacid strand without a standard nucleic acid template. How cool is that, guys? Is it interesting? Huh?
Yes.
Yes.
What about the length regulation of the produced
So
strand?
it depends. It depends, Yehan. So this is a mechanism being emerged, right? So there are multiple lengths of these segments made by the machinery, right?
Yeah.
Okay. So this is the fundamental interest here is that what made it the length depend on the template's length here at first part, right? The DRT3 base mechanism. So it doesn't need a template nucleotide. One second. It does not need a template nucleotide to make a polyacid stretch, right?
Yeah.
That is to make a double-stranded DNA. Now, this is what do you guys think as molecular biology students? Huh? There's a protein, there's a photocopy machine, make copies without inserting an original. The machine itself acts as a 3D printer for a new DNA segment. And it is intelligent enough to understand what sort of complementary strand that it needs to make in order to pair with what made by DRT3A. Okay. The protein, DRT3B protein as an enzyme, without having the first time in the history, a molecule, an enzyme showcase their ability to make a nucleotide molecule in template independent manner. The protein's active site itself can make a new DNA molecules by using incoming nucleotide without retrieving information from a template. So isn't it cool, guys? The protein as an enzyme, so it doesn't need the support of a template to understand what to make, right? How it happens? The active sites itself do not allow other nucleotide to enter and bind based on the certain amino acid residue arrangement on the active site of the protein. Because amino acid itself can do that function, it doesn't need the support of that template anymore to make a new stretch of DNA, right? So the protein has evolved into another level that they can make DNA. Can you guys feel the how scary these molecular evolution mechanism, where those mechanisms could direct into? The proteins start making DNA now, right? Sanjula?
Yes, sir.
Himal explained it before.
Sir, I have a question actually. May
Yeah, ask
like,
the question.
like it seems to be complementary, no? The poly-GT strand and the poly-AC strand seems to be
Yes,
complementary and make the double-stranded DNA. So
yeah.
how DRT3B knows to adding the next nucleotide without? So it is it's clear that its active site has the best shapes for the AOC, not GOT, but how they make the add the next nucleotide like complementary to the strand made by DR3A.
Yeah, it says according to what we learned so far, Himal, let's say now I don't know, I can't explain this. Let's say this is the DRT3B's active site, right? So I'll draw something and show you here. Can you see it? So let's say this pocket is the active site, right? Can you see it on my screen,
Yes, sir.
right? Now, initially this is DRT3B component, right? There's DRT3A component with the support of another non-coding nucleotide. It makes DRT3B here, DRT3A with the support of another nucleotide, it makes that poly-GT. Poly-GT part, this I don't know how. Poly-GT is made here. GT, GT, GT. It's made here, right? And at GRTB, there's no non-coding, but the nucleotides enters here, the pocket, right? It has to make AC, 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 AC stretch out of this. Okay. So your problem, Himal, your question is that how it figures out, right? How to alternatively attach it. Is that your question?
Yes, yes. That means how the DR3B knows the sequence to
That's
be.
that's what, yeah, interesting here. Now, normally in here, it's got the template. Always it can refer back to the template and see. I should add this, I should add this because the complementary one is this. In here, it does not have that reference, right? Then 14 amino acid itself should be arranged. Let's say asparagine and other amino acid, there are such amino acid. They are still investigating how the amino acid in this pocket can allow only A after T. One A is attached, the next interaction will not allow any other bases to come but T, based on the shape of the pocket. Okay. It's like, it's a 3D
Okay.
structure. When A enters, next T only can come until it folds back. Make sense? It's more like a scaffold. Only 80-80 order, the nucleotides can enter. Do you understand?
That means actually the DR3B active site itself determining the sequence like that.
It's an intelligent active site. Very
Yeah.
interesting, right?
Yes.
Now, the students who are molecular biologists, I mean, should become fascinated by what this protein does here, right? So you can see the potential of proteins to evolve into a stage in future that they can make even multiple at this particular point with the discovery. It is intelligent enough to make an alternative AC segment, right? Stretch of DNA. But now you can see, now you know that protein can evolve active sites to make DNA without the support of DNA templates. Can you see the potential of proteins to evolve into a stage that they can even make longer stretches? If you can engineer a protein with certain amino acid order, we are not there yet, but in future, if you can engineer amino acid order in a way that it allows only certain order of nucleotide to enter into it. So they will act as proteins that make DNA without the support of templates, right? So these mechanisms, now if you write this mechanism as a DNA synthesizing mechanism even in your answer scripts, would you get marks, guys? Tell me. Everybody? Huh? In your molecular biology papers, exam papers, you write this mechanism, the pocket itself make DNA with no templates, right? Sanjula?
Never, sir.
Never. We are at this stage, right? But can you understand that these kind of mechanisms can be evolved in biological systems? There are no rules in biological systems as long as it supports their survival, right? So you can be creative as well in understanding biological systems and in creating new biological systems. So that's why sometimes I tell my students, if I tell you amino acid can make protein. Remember, before this discovery, I mentioned this once. If I tell amino acid can make proteins, make DNA, you should believe it. Because sometimes now it happens in biological systems because they evolve, right? Many different kind of interesting mechanisms. This is a huge gap. Now, all the previous mechanisms are restricted into the template dependent aspect. But this finding shows no, protein can also make DNA with no template support. So it gives me goosebumps when fundamental goosebumps about molecular mechanism. But when you think about molecular aspects, and when you understand that biological systems are always flexible, of course, this is not a big deal, but it's a very interesting breakthrough that we understand the proteins can also make DNA without the support of templates, right? And it base pairs. Now we want to investigate certain mechanisms behind the fundamental mechanisms behind this aspect. There are some gray areas now. How, as you ask, what is the actual nature of the pocket? Some says there are two amino acid involves that determine the shape of the scaffold, that it only allows AT in order to enter and escape the stretch of DNA, AC in the order, and something the entire. So this was done with cryopreserved electro, like the what do you call this electron microscopic techniques, right? There's something more to it's emerging, the actual shape of the pocket. But what it does, it confirm that it does not need a template to make DNA. So this is what I wanted to actually highlight here in this particular talk, uh, that biological systems does so many interesting things and it follows very interesting mechanism when they encounter different threats, guys. The idea is to survive, right? At molecularly as well as yeah, as well as at all organism level as well. Yeah. So it's a combination of classical and non-classical synthesis. DRT3, what it does, right? 3A RNA template dependent, 3B protein guided, right? So now you understand how interesting this mechanism. Okay. DRT7 is another protein that recently, within the same month, the scientists discovered another protein. I don't want to highlight the mechanism of DRT7 here because it's not very well established. It's not yet published. It was published, mentioned only in a preprint. Therefore, it is of course very interesting that it works as RNA independent manner, protein RNA independent, like a template independent manner, and it shows another mechanism, another upgrade, protein kinase dependent protein that is not peer reviewed. Protein primed of protein guided initiation of DNA generation, right? So later, once it published, you can read about and then understand how interesting, what directions these molecules evolve, right? So this is fundamentally very important when it comes to molecular biology. What interesting things these molecules do inside of our body. They are very intelligent at molecular level as well. Now, when you compare these components, DNA polymerase as one of the core molecules, right? So it template dependent DNA. Trimeric cut, yes, 3 prime N needed to incorporate a new molecule. The product DNA, it's a classical mechanism because it template dependent. RNA polymerase, it makes based on DNA. It does not need primers, it de novo. And product is RNA. Of course, it's classical because it template dependent. Reverse transcriptase, it's RNA, the template, primary guard, of course, yes. Double-stranded DNA is what you get, right, as the product. Again, classical because it template dependent. Now look at DRT3, DRT family. That's where this huge gap occurs, right? DRT3 template is non-coding RNA plus protein, right? System specific. GT-AC-DS stranded, which means the 3 system specific mean DRT3A component does need a template and the other component does not. Poly-GT-AC-DS DNA standards are made out of that multilobular structure, right? So it's an imaging whether to classify. It will be classified as non-classical because one component of it is the template independent. 14. It's like there's photocopy machine that makes copies without having an original inserted. There's no other way to explain it, guys. What this 14 does, DRT7, DRT family, 14 guided, 14 prime. So there are questions about that DRT7 because the paper is not yet published. So we have to wait until they publish the paper. We only have the preprint of it. Right, so those are interesting molecules. Why DRT3 and DRT7 are breakthroughs? So this expand the classical view of DNA synthesis mechanism. This is the fundamental core mechanism that we learn in molecular biology throughout the entire molecular biology. So we always believe that making nucleic acid is a template dependent mechanism, but we realize that it's not. It is, of course, it does not entirely violate, but it can be evolved as well into certain directions. DNA synthesis is not exclusively template directed by nucleic acid, right? Bacteria systems as innovation sources. Now, in bacteria, you can study this mechanism by studying anti-phage defense mechanism. We learn about CRISPR system, right? Now we come across this very interesting template independent DNA making mechanism. How cool as the bacteria, they are basically playing with their DNA to defend against the threats that they encounter, right? They are very, very creative. They can even alter their classical biological mechanisms in order to act against their threats, the bacteria. So you can think of bacteria as an evolutionary laboratory to study new molecular mechanism. So if you focus on that defense aspect, you can maybe someday come up with concepts, maybe to get a Nobel, right? Do you understand how valuable bacterial defense mechanisms as to understand fundamental molecular evolutionary mechanisms? Antiphage defense is a reservoir of undiscovered enzymatic mechanism. If you conduct a research in that area, there's high potential for you all to come across an entirely new mechanism which has not been observed before or examined before by scientists, right? In synthetic biology, one of the pet areas of ours, so protein-guided synthesis could inspire programmable DNA writing tools. We can come up with applied science tools like DNA write tools can be developed by programming the protein structure. That particular, we can develop basically 3D printers, protein 3D printers for DNA, right? Do you understand? The classical polymerases copy from nucleic acid to templates. There's these DRTs. It suggests that in specialized biological contexts, protein themselves can help guide DNA synthesis. This is why these discoveries are fundamentally exciting, I must say. So I see the potential of this mechanism, the discovery of these mechanisms to receive Nobel sooner or later. I can't guarantee because there's a lot of politics going on giving Nobel award as well as of any other area, guys. But this has that particular value, this particular discovery of protein's ability to make DNA when you look into the fundamentals aspect of molecular biology. So that's why I wanted this to be shared with our students as well who are interested in molecular biology and everything. That of course we know applied science matters, but to understand applied science we have to thoroughly study fundamentals and how fundamentally molecular systems behave and how flexible that otherwise you can't troubleshoot to certain applications, right? Now, do you guys want to join these discussions? As I am done, we can have a small chat on what we just understood about molecular mechanisms. You can be open-minded about. You can think of anything. You can suggest anything as long as so it can be evolved at certain point. What to think as a mechanism, right? Because there are no strict rules in biological systems. The bacteria doesn't know rules. It will only try to survive. Everybody, so I'm done with sharing this specific insight on molecular mechanisms. So you guys can have your idea as well, and the way we think about biological system, how important for us to follow up, keep track on fundamental molecular changes is important to link it with your applied aspects. As I know some students are biomedical science, biotechnology students, so we are focused on applied aspect, but without knowing how these molecules fundamentally changing, evolving, we can't adapt it into our applied aspects in molecular biology, synthetic biology, genetic engineering, as well as the recombinant DNA technology, I would say, right? So, yeah, this evening I wanted to, as I came across this article recently published, I thought of translating this knowledge real time to our students who are interested because so that's something lacking in our culture of science here that we don't discuss the real-time science happening in the world. So these science to be reached to our certain teaching platforms, it would take 20 to 30 years, right? Then at that time, many more mechanisms would have evolved. To understand, we should develop this culture of discussing real-time science, right? Now look at this, what we have in our mind about DNA synthesis has some other optional ways as well that you can. Now you write this answer as a secondary backup. So that's correct. Not the entire, it does not entirely violate the central dogma or anything, but it's there. Okay, everybody, Sanjula and everybody, Kavya, do you guys want to take over the discussion? Any question or anything on this? And then, right?
Yes, sir. I have a small question. So,
Oh,
yes, this is
right.
a good idea when like designing a gene circuit as well, when it's come to synthetic biology.
Okay.
So, what kind of application like that we can like I got some ideas now, but
Yeah,
what
I
kind
can
of
understand
applications have you
share
suggest?
their ideas. Now, here, look at this, Sanjana. Now, the DNA writing is an important application. Let's say we have the computational tools like EVO2, right? That is to come up with entirely new DNA stretches. Those are AI tools, right? So,
Yeah.
something predicted by that. Can you see the potential of coming up with a real world EVO2 robo,
Yes.
intelligent protein that can make their own DNA in future?
Yes.
Huh? Do you understand?
Yes, sir. Yes, sir. I can, and even like there are like CRISPR tools as well, like CRISPR sensing tools as well. But
Yes.
if this one works, it's really like we can.
Even the future scientists in synthetic biology can start
Yeah.
working towards the direction of developing small protein machinery that can make small stretches of DNA by altering the active site's protein. Himal, you are planning to work on protein engineering aspect. Can you see how multidimensional these topics are, the way they evolve?
Um, yes, sir. Actually, then, so when we think about me,
Hmm.
so if we can investigate the mechanism by which how the active site changes when the base is incoming, so then we can actually engineer like if we can investigate it properly, so we can actually engineer a protein to make our own DNA, which desired DNA without
Hmm.
needing a template like so we can replace the traditional PCR reactions. Uh, we can
Hmm.
directly add the NTPs, and so the protein can engineer to make the desired template for
So
us.
much of potential, energy wise, efficiency wise, entropy wise, right? How to make the mechanisms efficient, right? Sanjula, about yeah, your idea is also the CRISPR based one, right?
Yes,
Can
sir. Like we
yeah,
can like use it as even a memory. Like we can like
intelligent
print a
protein.
DNA.
Yeah, the memory. You can embed memory
Yes, sir.
proteins.
Yes, sir. I just thought about this as a like I'm into biosensor, so I just thought about this as a biosensor, how we can like use it inside a biosensor. That's
Yeah,
what I was thinking, sir.
you can actually, because it deals with information. The DNA is all about informations in biological systems, right? I mean, the nucleic acid, what it stores is that's our IT language, no? So ATGC basically does what it does. It stores it's the nature's language, coding language, right? Okay, Sanjula, am I correct? As an IT
Yes.
person, you
Yes, sir. Like
working.
ATGs is the natural coding language
Those
of
are, yeah. Now, it was known only by DNA before. Now it's been learned by protein, right?
Hmm.
So protein, it's like the now you can make robots, AI powered robots, which is intelligent.
Yes.
Do you understand? Protein could guide themselves
Yes, sir.
because it understand what DNA used to tell them before. Previously, they never understood. They were only practicing the mechanism of copy. What it says, they do. What it says, they do. We don't no longer need to, in order to make a small stretch of this GZ something, I don't need your guidance because I know that you have been telling that to me for decades. Let me at least make it that small stretch by my own at this specific stage. That's what DRT3B does. Am I correct, Himal? Himal.
Yes, sir.
That's what it does, right?
It takes the DNA like data storage into another level then.
Yeah, it's elevate. Sanjula and others.
Yes, sir. That's the thing. Like
ඒක
really interesting.
ඒක හරියට something like this Sanjula and everybody. අපි හිතමු කාව්‍ය is our host. හරි? කාව්‍ය is our host for this today's session. හරි අපි කාව්‍යයට හැමදාම script එක ලියලා දෙනවා කාව්‍ය you have to tell this. කාව්‍ය script එක බලාගෙන කියවනවා. කාව්‍ය script එක බලාගෙන කියවනවා. But at specific stage කාව්‍ය කියනවා මේ පොඩි කැල්ල මට පුළුවන් දැන් කියලා මට තේරෙනවා. Right? Do you understand? ඊළඟ එකේ මේ මට ඕක බලන්න ඕනේ නැහැ script එක. I can just tell it. I know that's what coming. Is that correct? How we are trying to understand how proteins learn this? Huh? Sanjula?
Yes, sir.
So those are the things going in my mind. The proteins behave at molecular levels, what our organism levels, what things happen as it. Because that intelligence is going down to certain stages throughout molecular systems, throughout biological systems. Okay. Anything else guys, that you guys want to discuss? Otherwise, we can wind up and share some news or anything you guys want to share with the molecular biology enthusiasts who joined today, right? Okay, Sanjula, over to you all now. Thank you very much, guys.
Okay, so before we wrap up, I would like to highlight that this initiative is going beyond today's session. So at BLI Think Lab, which is situated in Mala Bay, we are actively conducting the physical brainstorming sessions where we collaborate synthetic biology research projects, exchange ideas, and also exploring innovative solutions together. So in addition, we are excited to introduce our beginning scientist sessions at BLI Think Lab, where we aim to connect with and learn from the global scientists, bringing their experience and insight closer to our community. So this is just the beginning of many exciting things to come. And we encourage you to stay connected with us and be a part of this journey as we continue to rethink. So with that, we have come to the end of today's session. So to our audience, thank you so much for your participation and thank you for your active participation. For being such an engaging part of today's session. So have a wonderful evening, everyone, and we hope to see you again soon. Thank you.